Okay, cool. So we're back, and this is where we left off. Um, Example of dihybrid cross. Just treat a dihybrid cross as two monohybrid crosses and use your knowledge of statistics to answer the questions. So it's like two heterozygous phenotypes, and every Punnett square. So we have 1/4 RR, 1/4 YY gives 1/16 RRYY, then 1/4 RR with half of YY gives 1/8 of RRYY, big R, big Y, small y, a quarter of homozygous dominant alleles of R with a quarter homozygous recessive alleles of Y give you 1/16 of RRYY, and half of RR gives you 1/8 and 1/4 and 1/8, and these all should give you the total, like all of these accumulated should equal to 1, and I'll draw that later too in my own spare time. We have the term wild type, which is used to describe an allele that is most common in the population given no mutations. This looks like ++ when discussing knockout genes or mutations. So however, if you're forced to assume a genotype, always assume it's homozygous dominant. If you're forced to assume a genotype, you should assume it's homozygous dominant. Yeah, unless you want to figure out if it's a heterozygous dominant or, yeah. So again, wild type means no mutations. It's an allele that is most common in the population given no mutations, and it looks like ++. If your two or more events are independent of each other, and you need the likelihood that both of those events will occur, you multiply the likelihood that each event will occur times each other. IAM stands for independent and multiply. So yeah, you multiply the likelihood, the probability for each event times the other event. So that's how it works. These will be making much more sense when it's in practice. That's for independent events, right? If you want the probability that either one or the other of your event will occur add the probabilities for each of them to occur separately together. EOA stands for either or equals add. So if you want the probability that either one or the other of your events will occur, add the probabilities of each of them to occur separately together. Genes, a segment of DNA that codes for a protein. It's easy peasy. Euchromatin is a less highly condensed and thus more often transcribed type of chromatin. Diploid, a nucleus that contains a pair of each chromosome for each set of chromosomes. So you have 23 sets of chromosomes, and if you have a pair, you are a diploid. And if you had only one chromosome for each set, then you were a haploid. A pair of 23 CRM equals 46 total. CRM, I believe, here refers to chromosomes. An allele describes a variation of a gene. So what does describe a variation of a gene? It's an allele. An allele is a gene, but two different variations of the same gene are said to be alleles of each other. That's why you have a dominant allele and a recessive allele. For example, the gene for brown hair and blonde hair are both genes of a hair color trait, but they are alleles of each other. Cool. I was thinking of a question, but nervous system organization. What makes up our peripheral nervous system, or PNS? It's neurons, right? Nerves. The PNS is nervous tissue outside of brain and spinal cord, nerves. And the CNS is brain and spinal cord. What is the name for the part of your nervous system constituted by your brain and spinal cord? It's the central nervous system. What nervous system is further divided into parasympathetic and sympathetic? It is the autonomic nervous system, right? The PNS is further divided into the nerves that innervate skeletal muscles, or your blank nervous system, and the nerves that innervate smooth muscles, or your autonomic nervous system. So for skeletal muscles, because they're voluntary muscles, it is the somatic nervous system that innervates it. And what innervates these smooth muscles is the autonomic nervous system. The somatic nervous system is voluntary. You voluntarily flex your muscles to move or impress other people. Rest and Digest describes the parasympathetic nervous system. By far the most common distinction you'll have to make on the MCAT is between parasympathetic and sympathetic. Usually this will present as a question that gives you a scenario that you can categorize as fight or flight, or rest and digest. The trick is often that you'll have to pick a physiological response to the sympathetic or parasympathetic nervous system rather than just the terms themselves. Some common physiological responses to the sympathetic nervous system are pupil dilation, because you freak out, halt on GI peristalsis, you stop on your gastrointestinal peristalsis and digestion. You have increased blood to the skeletal muscles because you want to run faster, you want to just fight or flight. You have increased heart rate. You have inhibition of digestive enzymes, including saliva. So you want to avoid any digestion from both saliva and your GI tract. And you dilate your pupils, and you have increased blood to both heart and skeletal muscle and heart rate. And physiological responses to the parasympathetic nervous system are much the opposite, more GI peristalsis, digestive enzyme secretion. Your bronchioles and pupils constrict. You have a slower heart rate because you just want to chill and digest, rest and digest. So be able to link an intruder coming into your home with pupil dilation, salivary inhibition. You see here that your bronchioles also constrict when, interesting. You relax your bronchioles when you are in sympathetic. It's the reverse. That's the only thing that gets relaxed because you need to breathe more air when you are in a fight or flight mode. And you stimulate glucose production and release because you need energy to fight, right? For rest and digest, you need less energy and you need less air, so you constrict your bronchioles. And there's also the secretion of adrenaline and noradrenaline due to stress. You see that a lot of people having increased epinephrine and norepinephrine, and other names for adrenaline and noradrenaline, and you inhibit bladder contraction. So that's another thing. And you also stimulate orgasm when so when you're in sympathetic mode, like too much activation and stimulates orgasm. And however, in parasympathetic, you contract your bladders. You avoid getting pissy. In parasympathetic, instead of the glucose production and release, you have the bile release to help you with digestion, right? Same thing here. I forgot to set my timer. I'll put 120 for now. Okay, now we're in Newton's laws. So we have that net force is the sum of all forces acting on an object and their directions. Net force does not mean there are zero forces on the object. It just means those forces are balanced and acceleration is zero. For example, a car is on cruise control. The force of propulsion from gas combustion is balanced out by friction and air resistance. So when your car is on cruise control, because you're going at a steady speed, the friction from the road and air resistance is equal to the propulsion from the gas combustion. So they balance out each other. That's when you have a net force of zero. So the net force of zero does not mean that there are zero forces on the object. It means that those forces are balanced and the acceleration is zero. Newton's third law is that every force has an equal and opposite force. So if I push down on the floor to jump, the floor is also pushing back upwards on me. If it didn't, my foot would go straight through the floor. Makes sense, totally. So it was a tricky one back in the days for me, but the third law is basically that every force has an equal opposite force. This also really, like, the concept of you not going through the floor is about normal force, which is a reaction to gravity. So that's why the floor exists, and you can, like, that's why when you do not go through the floor, all I'm trying to say. The Newton's second law, the acceleration of an object is dependent upon its mass and the force applied to it. So F equals ma, this is probably the highest yield physics principle, which is Newton's second law, and that acceleration is dependent on mass and the force applied to it. And the first law of Newton is the law of inertia, which is a mass will remain at rest or in a straight line velocity unless acted upon it by an external force. This is why you must apply force to move an object and to stop an object that is in motion. Very rarely tested on the MCAT, but some keywords to look for are inertia and momentum. This is the law of inertia, which is the first law. If a velocity is constant, then its rate of change is zero, and its net force is zero. So, constant velocity equals zero rate of change and zero net force. Because if your velocity is constant, you have zero acceleration, and the net force is zero. Be on the lookout for the term constant velocity. They're trying to tell you that acceleration and force equals zero. Nice. We have thermodynamics here. Thermodynamics is a function of a molecule's stability and equilibrium. So thermodynamics is a molecule's stability, so it's a function, right? And that function represents a molecule's stability and equilibrium. Are these two different concepts? We'll figure out. When we speak in terms of a molecule's basicity, we are looking at thermodynamics. Is the conjugate acid very stable? Is the equilibrium shifted toward the conjugate acid? If so, then the species is considered a fairly strong base. So a molecule's basicity is when we are looking at thermodynamics. Is the conjugate acid very stable? Equilibrium shifted towards the conjugate acid. Then the species is considered fairly strong base. So if the conjugate acid is very stable, like for the amino acid, the carboxylic acid in the proteins, if you want to see if the base is strong, then should be at equilibrium, right? Is the equilibrium shifted towards the conjugate acid? If yes, and the conjugate acid is stable, very stable, then you know that it's a strong base. So the strong base leads to a stable conjugate acid. An electrophile is a chemical species that acts as an electron acceptor, so you want electrons. So phile means that it just loves electrons, so he wants it. It's attracted to electron. And nucleophile loves the nucleus, like protons, right? So it's a electron donor. It's a chemical species that acts as an electron donor. Now we have these. Oh, the lovely SN2 SN1 elimination substitution reactions chart. We have the methyl group, primary, secondary, and tertiary substrates. We have polar protic solvent, polar aprotic solvent. We have strong small base. We have strong bulky base. Knowing what each of these solvents are and like what kind of base it is, that is, I think, the tricky part, and you can only practice it with doing questions on it. So we have E1 and E2. So bulky base, small base, strong small base, or strong bulky base, and we have polar protic and aprotic. We need to know what, I forgot what protic is and aprotic is. Yeah, you can see that these ones are unlocked because I've done them before. I did them a few weeks ago on the plane when I was going to Germany. And we have the variable ratio is what is reinforcement after random number of successful attempts. So because it's random, and it's reinforcement. Because it's random, it's variable, right? And because it's attempts, number of attempts, it's a ratio. Random number of successful attempts. If it wanted to be an interval, then it should be about a time period, right? And these are all about operant conditioning, which is different from classical conditioning. Operant is basically reward and punishment, right? You want to reinforce a behavior or just prevent it. So if you want to reinforce a behavior, you need to provide rewards for that. And we have the variable interval, so it's reinforcement after random time periods. We have positive punishment, which is adding a stimulus to decrease a behavior. So because it's a punishment, because it's a punishment, you want to reduce a behavior, decrease a behavior, you want to avoid that behavior. And because it's positive, you're adding something. So you're adding a stimulus, like pinching your brother because he played a prank on you. You don't want to see that prank anymore, just pinch him. The positive aspect means the stimulus was added, so the pinch, and trying to decrease the likelihood of your brother playing pranks on you is a punishment. Now we have operant conditioning, which runs on the premise that you can reinforce a behavior to increase the likelihood of that behavior happening again. The whole chapter is explained in this YouTube video, apparently. You can watch that later. I have not watched it. We have negative punishment, which is removing a stimulus to decrease a behavior. And for example, taking away your child's keys after you caught them vandalizing the city. The negative aspect means there was a stimulus taken away, so it's a punishment. The negative means a stimulus was taken away, so you take away the keys. And the punishment is decreasing the likelihood of your child vandalizing a city, so you punish him. A fixed ratio is ratio is about the number of successful attempts, and fixed means that it is certain. It's after a set number of successful attempts. We need examples of these for sure. Fixed intervals, so it's reinforcement after set time period. And something being positive in the realm of operant conditioning means that there was a stimulus presented. Something being negative means that a stimulus was taken away. So when something positive is, when you say positive, it means that something was added, presented. And negative means that taken away, simple. And positive reinforcement is like adding a stimulus to increase a behavior. For example, giving your employee a raise because they are doing such good work. The positive aspect means there was a stimulus presented, in this case money, and we are trying to increase the likelihood of that employee continuing their good work. So it is a reinforcement. So yeah, you just increase their salary. A negative reinforcement is just removing something bad to increase that behavior. For example, removing a strict curfew from your child since they made all A's this semester. The negative aspect means that there was a stimulus taken away, the strict curfew, and you want to increase the likelihood of the child making good grades. It is a reinforcement. Oh, so many organelle transport cards here. Okay, let's do it. We have simple diffusion. So free diffusion down a concentration gradient. So when you are experiencing a free diffusion, okay, nice. The plasma membrane is permeable to some molecules which can freely go into and out of the cell, depending on the concentration gradient. Usually the only molecules that can do this are atomically small and hydrophobic like O2 gas. Charged ions like Na+ are hydrophilic and thus cannot diffuse across the membrane. The plasma membrane is permeable, we know that, which means it is permeable to some molecules, which means those molecules can freely go into and out of the cell depending on the concentration gradient. Usually the only molecule that can do this are atomically small and hydrophobic like O2 gas. So we know that you should be hydrophobic in order to pass through the cell membrane, because these heads are hydrophobic. In order to pass through them, you need to be hydrophobic. My apologies. The cell membrane itself is a bilayer of phospholipids, right? And however, these heads are hydrophilic. But in order to pass through this membrane, you need to be hydrophobic, because you need to pass through it, right? The inside, the middle of the cell membrane is literally hydrophobic. So charged ions like Na+ are hydrophilic and thus cannot diffuse across the membrane. Like Na+ cannot pass through it. So it cannot have simple diffusion. So simple diffusion is just outside and they pass through it. A facilitated diffusion is different. These are also passive transports. You don't need to use ATP for this, right? So you have transported substance, you have transporters, and these transporters transport something like glucose via facilitated diffusion. You also have osmosis, for which you have aquaporins, and the water molecules just go through the aquaporins for that. Okay, we have active transport, which uses the energy from one gradient A, to drive movement of a separate molecule against its gradient B. Does not directly use ATP, although gradient A can be created by using ATP in primary active transport. So you have primary active transport, which is when you are increasing the concentration of, for example, sodium, and you want to bring the Na+ from the outside to the inside of the cell. You will use ATP, and this is an example of antiport, where with each sodium that comes in, there are potassiums coming out. There is also the opposite, the symport. So when you're bringing out sodium from the cell, when you're bringing it outside the cell, you have glucose, where you also bring that out. So that's a second reactive transport, and it's a symport. So does not directly use ATP, although gradient A can be. So secondary active transport does not directly use ATP, although gradient A can be created by using ATP in primary active transport. So again, second reactive transport is using the energy from one gradient A, to drive movement of a separate molecule against its gradient B. Oh, this is all second reactive transport. It doesn't directly use ATP. Okay, so when in this right example, if you're using another substance, another gradient, to drive movement of a separate molecule against its gradient, that's when you're doing secondary active transport. It doesn't use ATP. That's the key point. If you're using ATP, then you're doing primary active transport. See here, when you're using ATP to directly drive movement of a substance against its concentration gradient, it's always about going against your concentration gradient, then you're doing primary active transport. One molecule moves against its gradient. There might be some confusion here, so we can ask what's the topic? Organelles transport. Organelles transport. Are antiport and symport part of primary or secondary active transport? In other words, does an active transport is it the primary or secondary active transport? That is it primary or secondary active transport that transports the molecule it's opposite gradient, right? Against its gradient. Concentration gradients. We also have osmosis, so we figured out that that happens with aquaporins. So osmosis is the passive diffusion of water across the plasma membrane. This can occur via simple diffusion or through a protein channel, like aquaporins, in a facilitated diffusion like manner. Cool. What is a unique organelle that contains its own DNA, which is inherited maternally. Ooh, maternally. It means that mitochondria comes from our mothers and is inherited maternally. So you have your mother's mitochondria only and not your father's. Like the DNA, right? And the mitochondrial DNA diseases can only be inherited from your mother. Now, a follow-up question would be, what are the most common mitochondrial diseases? It should be related to our metabolism, right? Well, how are mitochondrial and mitochondrial DNA diseases different? Examples, please. We have active transport again, using energy from ATP to move substances against their concentration gradient. I'm starting to believe that it should be against the concentration gradient in order to be active transport, but I don't know why there's also a symport. Usually the only molecules that can use simple diffusion are atomically small and hydrophobic, like O2 gas. Yeah, we know that O2 gas is atomically small and also it is hydrophobic, so it can pass easily through the cell membrane with simple diffusion, passive diffusion. Charged ions like Na+, sodium are hydrophilic and thus cannot diffuse across the membrane. Typically, proteins are synthesized by a ribosome, which undergo post-translational modification in the RER, that is the rough endoplasmic reticulum. So, and then go to the Golgi apparatus to be packaged in exocytosed outside the cell and to its target elsewhere in the body. This is the diagram. So proteins are synthesized by ribosomes. So we must know that ribosomes are literally what make the proteins. So whenever you're asked to think what synthesizes proteins, that is the ribosomes. We know also for a fact that when the tRNA is translating the genetic code and attaching all those amino acids together to make up the protein, though that takes place in the ribosome. The ribosome comes there and just protects the whole thing and make it happen. And the post-translational modification happens in the rough endoplasmic reticulum. So it is in the RER, and it is rough because of the proteins. The soft endoplasmic reticulum, is it soft? Is it called soft or is it called something else? Smooth, sorry. The smooth endoplasmic reticulum consists of lipids, right? And lipids are soft. Like baby bum is just soft, right? Because it's fatty and like a fat ass. I had to say that, sorry. And then they go to the Golgi apparatus to be packaged. So yeah, the ribosome synthesizes the proteins. The post-translational modification takes place in the rough endoplasmic reticulum, and then you send it to the Golgi apparatus to package it in, exocytose it in outside the cell, and send it to its target elsewhere in the body. You have rough ER, you can see, and the vesicles transport to Golgi apparatus. The Golgi apparatus has three, like there are like two main hypotheses about Golgi apparatus. One of them is the arrival section, and then there's the middle section, which is the cisternae. I hope I'm not confusing that with the mitochondria. Definitely need to get these facts straight. And then there is also the exit section of the Golgi apparatus. How is Golgi apparatus structured? What are the two hypotheses about its function? Yeah, that should help. It's somewhere in the flashcards too. I've seen it, but regardless. So yeah, same thing. We have the smooth endoplasmic reticulum, smooth ER, which is continuous with the rough ER, and does not have ribosomes embedded in it. This is what makes it smooth, and it functions to synthesize lipids and some hormones and detoxifies harmful chemicals. There's also a subtle difference between the soft endoplasmic reticulum, where it is detoxifying harmful chemicals, and also the peroxisomes. And there is also something else than peroxisomes. We'll get to it. I've seen it somewhere, I remember. And yeah, so the main thing about smooth endoplasmic reticulum is that it doesn't have ribosomes. It's continuous, however, with the rough ER, and it functions to make lipids, synthesize lipids. It also synthesizes some hormones, and also detoxifies harmful chemicals. So what are the organelles in the cell that detoxify harmful chemicals? One of them has to do with like hydrogen peroxide. Yeah, peroxisomes do that. For later, rough endoplasmic reticulum continues with the nuclear envelope, and soft ER, smooth ER, it contains ribosomes, which is what gives it the rough appearance. It can technically be considered a site of protein synthesis because it contains ribosomes. So yeah, rough ER is continuous with nuclear envelope, and at the end you have the smooth ER, right? So first is rough and then smooth, and it contains ribosomes, where literally you synthesize proteins. The ribosomes give it the rough appearance, and beginning side of post-translational modification. So they start post-translational modification. Once this process is begun, the protein is transported to the Golgi apparatus to finish. So the finishing touches are are fulfilled by the Golgi apparatus. And what else? We have ribosomes, and they're a site of translation, sure, and they can be free floating or embedded in RAW-VR. Embedded in RAW-VR, sure. What does it mean that they can be free floating or embedded in RAW-VR? So they just free float in the do ribosomes just free float in the cytosol? That is the space surrounding the organelles inside the cell. Proteins synthesized in free floating versus RER have different destinations and jobs. That is, what difference do ribosomes what difference do the proteins from free floating versus RER? Polarity of a phospholipid. The phosphate head is hydrophilic and the fatty acid tail is hydrophobic. Totally makes sense. That's why when one of these is upside and the other one is downside, in the middle it is hydrophobic. So it's fatty acid tails, two fatty acid tails. If it is saturated, it is unbroken. If it is unsaturated, it is broken. So the double bond means that it is unsaturated. And if you saturate that double bond, it turns out to be a straight tail. But if it is a double bond, which is unsaturated, the saturation is basically turning the double bond into a single bond. Then if it's unsaturated, it has a tint, like tilt. What do you call it? Kink. Sorry, my brain is fried. Hey, but we have to get through this. You lick with your tongue, which is on your head. Let me sip some water. Like anyone watching these recordings must have gone crazy by now. A bit of crazy is good. We have the hydrophobic head. Nice. This is the phosphate. And this is the glycerol. Phosphate is PO4, PO3. But if it's PO4, it means that it's minus. So the phosphate head is hydrophobic, and the fatty acetal is hydrophobic is the fat. Yeah, you get it. This is about the polarity of a phospholipid. You lick with your tongue, which is on your head. What does it have to do with hydrophobic and hydrophilic? Oh, hydrophilic. You lick with your tongue, which is on your head. No, that doesn't do it. It's easy to understand because hydrophobic is basically that's what it is, touching the water, the liquid outside or inside the cell. You just have to picture the structure of the cell membrane in your head and you're done. Boom. The plasma membrane proteins function as receptors and channels. The plasma membrane proteins function as receptors and channels. Sure, you can have either receptors or channels. Those are proteins, and they are inside the plasma membrane. The plasma membrane phospholipids consist of fatty acetals attached to a glycerol backbone, which is attached to a phosphate head. So the phospholipids inside the plasma membrane, they consist of three things. The fatty acetal, which is hydrophobic, and it's two. There is the head, which is hydrophilic. The head consists of glycerol and phosphate. Glycerol backbone. So the fatty acetal is attached to a glycerol backbone, which is attached to a phosphate head. Plasma membrane lipids are generally cholesterol, which functions to increase or decrease fluidity. So the lipids inside the plasma membrane are generally cholesterol, which function to increase or decrease fluidity. We need to understand what is fluidity. At high temperatures, cholesterol decreases fluidity. At low temperature, it increases. There are several lipids in the plasma membrane. Cholesterol is just the main one for the MCAT. What is fluidity? And how does cholesterol control it in the plasma membrane? The plasma membrane composition. It is composed of phospholipids, proteins, lipids, and carbohydrates. Oh, we have all of them all together. We have phospholipids, cholesterol, which is a kind of a lipid, and carbohydrates and proteins. Plasma membrane carbohydrates function as signaling molecules and cell recognition. So now we understand also the function of carbohydrates, where they're used in signaling or cell recognition, kind of like a work badge that allows you indoors and to be recognized by other employees. Good. I like the analogy. No, I say good analogy. How are carbohydrates how do carbohydrates in plasma membrane help with signaling and cell recognition? What are some examples? Ooh, phospholipids are amphiphilic. Amphi, meaning they have a portion that is hydrophilic and a portion that is hydrophobic. Huh. So amphiphilic means amphibian. Oh, for example, amphibians, they can live in both water and on soil. So so amphiphilic means that it's both hydrophobic and hydrophilic. Oh, here it is, peroxisomes. They utilize redox enzymes to neutralize free radicals, which helps break down fatty acids, amino acids, and detoxify chemicals. So peroxisomes help in breaking down fatty acids, amino acids, and detoxify chemicals. How? By utilizing redox enzymes. There was one specific one, I think it was catalase, that was specifically for hydrogen peroxide and the radical oxygen. That was, oh yeah, it was also related to the aerotolerance microbes and all of those 5 different ones, like facultative, aerotolerant, anaerobic microbes. So they create hydrogen peroxide in the process. That's a byproduct. Hydrogen peroxide is less dangerous than the oxygen, radical oxygen, like the superoxide. We can definitely learn more about that. To neutralize free radicals. So you neutralize free radicals. Like superoxide is neutralized. You need to neutralize them because they have a charge, a free radical, I believe. Like one more proton, or how are free radicals dangerous? Do they have an extra molecule? What do you call it? Molecule or atom? Or electron? Superoxide example, where it is also taken neutralized by, is it neutralized? Yeah, by peroxisome. Okay. Also, what is the redox enzyme? What is the redox enzyme that basically helps peroxisome? Peroxisome. Is it catalase? I'm being smarty pants, eh? Again, we have passive transport that does not use ATP. The nucleus is the site of DNA replication, DNA transcription, post-translational modification, post-transcriptional modification. We know that it was the rough ER that was responsible for post-translational modification, starting it, and it finishes in the Golgi apparatus. But here, be careful. There is also post-transcriptional modification, where it happens in the nucleus. And nucleus is a site of DNA replication, DNA transcription, where that's where you want to make a copy of the DNA so you can later on translate it into a protein. And some post-transcriptional modifications also take place in the nucleus. The nucleus is surrounded by a nuclear envelope with two phospholipid bilayers called the inner and outer membranes. Oh, nice. So the nucleus is surrounded by a nuclear envelope. We need a picture of this to understand it fully. Nuclear envelope. So it separates the contents from nice 3D model. I like that. I didn't know Google had that. So it separates the contents of the nucleus from cytoplasm and provides the structural framework of the nucleus. So yeah, you have the nucleolus, that's the inside of the nucleus. Nuclear envelope, you have the outer membrane, the inner membrane. You have nucleoplasm, which is the space, the environment between the nucleolus and the nucleus, like the nuclear envelope. You have the chromatins. The heterochromatins are more condensed. You can see them, but euchromatins are expanded, so you cannot see them. It seems that it's closer to, like it's attached to the nucleolus, but we will understand that later. And you have ribosomes, you can see here, and nuclear pores. Nice. This is actually, let me put this in here. Here we go. Why is it 2 images? You only need 1. The nucleus also contains the nucleolus, which is where ribosomal RNA and therefore ribosome synthesis occur. So you also need to make ribosome, right? You remember that ribosome was making proteins. Now you need to keep in mind that you need to also make the ribosome. So nucleolus is where the ribosomal RNA and so the ribosomal RNA is there and the ribosome is synthesized. We now need to know how is ribosome synthesized in the nucleolus. from rRNA. And how is rRNA unique? The mRNA transcripts exit the nucleus through small holes in the nuclear envelope called nuclear pores. So mRNA transcripts exit the nucleus through small holes in the nuclear envelope called the nuclear pores. Oh, here you can see cisternae. It's around the nucleus, right? And so again, the mRNA transcripts. So what are mRNA transcripts? We know that, okay, it should mean that the mRNA genes, right? They exit the nucleus through small holes in the nuclear envelope. Oh, so you can see the mRNA exiting the nucleus through the small holes called nuclear pores. So they come out of the nuclear pores, they're holes, small holes in the nuclear envelope. Envelope is the membrane, right? The two phospholipid bilayers. Biphospholipid layers. Let's see. Phospholipid bilayers. The two phospholipid bilayers. Nice. Is the mRNA transcript the same as mRNA? What does the M stand for, the mRNA? Is it mitochondrial RNA? Okay. We have the mitochondria. We all know that it is the powerhouse of the cell. It is composed of an outer membrane, inner mitochondrial membrane, cristae, foldings of the inner mitochondrial membrane, and mitochondrial matrix, space that is surrounded by the inner membrane. So you have an outer and an inner mitochondrial membrane. And then you have cristae, which are the foldings of the inner mitochondrial membrane, and the mitochondrial matrix, which is a space inside the inner membrane. The mitochondria also maintains a C3 proton. The proton. It maintains a proton gradient across the inner mitochondrial membrane, which is used to drive ATP synthesis. So there is a gradient, which is a proton gradient, and it is across the inner mitochondrial membrane, which is used to drive ATP synthesis. If this gradient is disrupted, there is no energy available to create ATP with. There is no energy available to create ATP with. You cannot make ATP if this inner mitochondrial membrane is disrupted. This is a common MCAT question. Let's take a look at the mitochondria. See if we can get this cool 3D model as well. Yeah, we have it. Nice, you can zoom on it. This is membrane, ribosomes, the matrix, cristae is the inner membrane of each mitochondrion invaginates to the inner membrane of each mitochondrion invaginates to form a series of folds called cristae. So the cristae are the folds that are the folds of the inner membrane of the mitochondrion. It greatly increases the surface area of the inner membrane and create multiple compartments in the fluid-filled inner cavity known as the matrix. It creates multiple compartments. So we can ask, what compartments does cristae make inside in the matrix of mitochondria? These are based questions. Maintain yeah, so this is also a question that I need to ask for myself. What is the proton in the proton gradient that exists at the inner membrane of mitochondria? That is involved in ATP generation, right? Okay, cool. I can write from here too. Yeah, it is the inner mitochondrial membrane. We understand all of that. It's two membranes, outer membrane and inner membrane. Here it is, lysosomes. So we had peroxisomes and lysosomes. We asked a question here, right? I.e. lysosomes, peroxisomes, so we want to know all the similarities and differences, right? Versus peroxisomes. There's another one too, right? Lysosomes come from lysis, right? They contain hydrolytic enzymes. So, right here, peroxisomes, they contained redox enzymes, but lysosomes contain hydrolytic enzymes. So, what is the difference between hydrolytic and what's the difference between hydrolytic and redox enzymes? Examples, please. So lysosomes contain hydrolytic enzymes and an acidic pH, so they can break down foreign particles or unneeded macromolecules within the cell. They're the cell's garbage disposal. So when you're a garbage disposal, it means that all the trash goes into you, right? And you contain hydrolytic enzymes. It's like a fancy garbage disposal. It's like having a trash bin and a lid on it, and the trash bin, like the garbage, like the trash bin is able to detoxify the garbage with hydrolytic enzymes, and also having acidic pH, so it can break down the garbage, the foreign particles or unneeded macromolecules within the cell. We can also have some examples here. So I hope you can see, yeah, what are the examples of macromolecules. What are the examples of macromolecules that lysosomes unneeded macromolecules that lysosomes break down and get rid of? Oh, foreign particles too. Okay. Lysosomes are also a common organelle for disease processes, creating a class of diseases called lysosomal storage disorders. For this reason, you may encounter passages discussing lysosomes in depth. So lysosomes are also a common organelle for disease processes, creating a class of diseases called lysosomal storage disorders. So lysosomes are also a common organelle for disease processes, creating a class of diseases called lysosomal storage disorders. What are lysosomal storage disorders? And give me examples. Golgi apparatus. It modifies, sorts, and packages proteins before shipping them to where they need to be. So we have also the Golgi apparatus. We know that it modifies, sorts, and packages proteins before shipping them to where they need to be. Think of the Golgi apparatus as the post office. Typically, proteins are synthesized by a ribosome, undergo post-translational modification in the rough endoplasmic reticulum, then go to the Golgi apparatus to be packaged in exocytosed outside the cell into its target elsewhere in the body. Is there any other place than Golgi apparatus where proteins are modified, sorted, and packaged? Elsewhere in the body. What are examples? Of substances of proteins. Examples of proteins that what are examples of proteins that go through this Golgi apparatus? Peptide hormones, perhaps? Facilitated diffusion. It does not use ATP but uses chemical gradient as its energy source to diffuse across a protein channel. Does it have to be protein channel? Does it have to be protein channel for facilitated diffusion? To transport does it have to be protein channel for facilitated diffusion? It uses chemical gradients for transporting. What does it transport? Facilitated diffusion. Ions or big molecules. Examples. We need examples always. Beginning side of post-translational modification is the rough endoplasmic reticulum. The end site is Golgi apparatus. At high temperatures, cholesterol decreases fluidity. At low temperatures, it increases fluidity. A concentration gradient is a driving force on particles to reach equilibrium. So A concentration gradient is a driving force on particles to reach equilibrium. This occurs when the concentration of particles in one area, say across a semi-permeable membrane, like the plasma membrane, is higher than the concentration of particles on the opposite side. So what is the driving force on particles to reach equilibrium? It's all about concentration. Like, it goes back to Le Chatelier's principle, right? So what was the question I wanted to ask? Nothing, I guess. Yeah, so the driving force on particles to reach equilibrium is concentration gradient. Nice. That was like 56 cards. Let's say half of it was concepts, so it was like 25 different concepts. It took us a while. We also have the ionization energy. We're now in the periodic table trends. So we're going to just finish this in 2 minutes and boom, done. We have ionization energy, which is the amount of energy needed to remove one electron from the valence shell of an atom. So each atom has a valence shell, and in order to remove an electron from that, it's called the ionization energy. It increases as you go to the right and up of the periodic table. We have it. So like, I'll show you that later. We have that. It's in the first page. It's the first thing you notice in this document. This is the second page. My bad. Ionization energy, it increases as you go right and up. Nice. Next. Electronegativity, the tendency for an atom to attract or hold on to shared electrons. If you want to hold on to your electrons, and you love electronegativity, you want to attract or hold on to your shared electrons. It increases as you go up and right. Makes sense, because the more electrons you have around you, you just want to hold on to them. You don't want to share them a lot. The less you have, you just want to give them away, right? The atomic radius describes the distance from the nucleus to the outermost shell. The atomic radius increases as you go down and to the left. On a periodic table, the electronegativity of an atom increases as you go up and to the right. As you go to the right, your nucleus increases in size. Yes, meaning it has a larger positive charge. And as you go up, you decrease the number of valence electron shells you have. This allows the nucleus to be closer to shared electrons, and thus exhibit a stronger pull on the incoming shared electrons. So our time is also up. I'm just gonna finish these, and so we only have this much left to complete the overview. However, I might need to go back on these, as I'm not sure if I fully went over them yesterday. So yeah, then we have a good grasp on the high yield material in only a day or two. That's like it's only 3-4 hours that we have been working on this, and then we can just like get into the details and get more and more completed, done. So electronegativity of an atom increases as you go up and to the right, because as you go more up, you have a smaller atom, and your nucleus is closer to, you have less valence shell, so your nucleus is closer to the valence shell, and also when you go to the right, you have more electrons, so you are attracted to more of them. So as you go to the right, your nucleus increases in size. That's interesting. Your nucleus increases in size because you have more protons, and at the same time, meaning that it has a larger positive charge. So your electronegativity, your attraction to your electrons goes up. It's your tendency as an atom to attract or hold on to shared electrons. Ionization energy increases as you go up and to the right again, because ionization energy is the amount of energy that you need to remove one electron from the valence shell. So it's harder and harder if you go to the up and right to remove an electron. And in an ionic bond, in what kind of bond? One molecule donates its electron and one takes that electron. So when one molecule donates its electron and the other one takes that electron, it is the ionic bond. This creates an electrostatic attraction between the two atoms because they know that they need each other. That electrostatic attraction is the bond, right? Electrostatic attraction. What is an electrostatic attraction? It's like we are in the periodic table trends. What is an electro go away. What is an electrostatic attraction? Electrostatic attraction is between the two atoms because they know that they need each other. The bond is the electrostatic attraction. And what kind of bond is where the electron is shared between the two atoms? So when the bond is one of them giving the electron is electrons, like the ionic bond. One molecule donates its electron and the other one takes it. It's ionic. When they share the electrons, it's covalent. The electron is covalent, meaning it's in both valence shells. Oh, covalent. Valent comes from the valence shell, and it's in both valence shells. The atomic radius should be increasing. Also when you go, it's the opposite. When you go down and to the left, the further you go down, the more electron shells you have. That is correct. So the further to the left you go, the less protons you have. So if you have fewer protons, creating a positive charge to pull on the electrons in the outer shell, those electrons in the outer shell are going to be at a further distance from the nucleus. So the atomic radius is different than the nucleus, than the nucleus size, the nuclear size. Where was it? The atomic radius. Electronegativity. Your nucleus increases in size. Yeah, so the nucleus size is actually kind of like the opposite to the atomic size. So because your nucleus is increasing, like the lower you go, let's check here. So the lower you go, it means that there are more shells, right? So that's when your atomic radius is increasing, right? And the more you go to the left, it means that you have less electrons in your valence shell, right? And also less protons. So when you have less protons, the force of the proton is, let's ask it, is the force of the proton much stronger than the force of the electron? I believe so. Hell yeah. So because the force of the so you are, when you are going to the left, you are reducing, you're lowering in the number of your protons and electrons one by one, right? But the force is exponentially going lower. So you're having less protons when you go into the left compared to, and your valence shell is decreasing. So less force. And when you're going to the left, it means your atomic radius should decrease, no? So the further down you go, the more electron shells you have. Right. That's right. You have more electron shells. No denial in that. And because you have more electron shells, your atomic radius just increases. So the further to the left you go, the less protons you have. Wait, the further left you go, the less protons you have. Right. So if you have fewer protons, creating a positive charge to pull on the electrons in the outer shell, those electrons in the outer shell are going to be at a further distance from the nucleus. What? Those electrons in the outer shell. So if you have fewer protons, creating a positive charge to pull on the... Oh, when you're going to the left, you have fewer protons to pull on it. That's why you are increasing your in size, because you have just few protons. You have fewer protons to pull on the electron. You have to see this as the attraction. So the nucleus is pulling. It's not pushing, right? So the more it's pulling the electrons, it means that the atomic radius is lower. But when you have fewer protons that go to the left of the periodic table, it totally makes sense. You have fewer protons, so the atomic radius increases. Fantastic. And also the metallic character just goes up as you go to the down and left, and non-metallic character goes up. So ionization energy, electronegativity, they all electric run affinity, whatever that is. Electronegativity, ionization energy. So what is the difference between electron affinity and ionization energy? Is the energy you need to remove that valence shell electron, and electron affinity is just like the affinity that your nucleus has to. So again, because you're going to the right, you have more valence shell and you have more protons. So because you have more protons, it is attracting more and more of the electrons. So it is harder. It requires more ionization energy to remove that valence shell electron. Again, the electronegativity is also increasing because the protons are higher and higher as you go to up and right. And up is less valence shell electrons. So even stronger pull force from the nucleus. Amazing. We did this.